Good afternoon. Welcome to the 20th anniversary season of the Congressional Biomedical Research Caucus. I'd like to begin by thanking caucus co-chairs Congressman Brian Bilbray, Mike Castle, Jackie Spears, and Rush Holt, and their staffs for their commitment and dedication to the ongoing support of the caucus. Without them, these caucuses could not take place. And I'd also like to thank the Howard Hughes Medical Institute for its support of the caucuses through a very generous grant. These briefings would not take place if it was not for the support of HHMI. I want to remind you that you can find past briefings on the CLS website at www.coalitionforlifesciences.org. And there you can also register for an RSS feed to get briefings about future postings. Today I'd like to introduce Dr. Henry Chambers from the University of California, San Francisco. Dr. Chambers has been a member of the medical faculty of UCSF since 1985. He is currently the Chief of Infectious Disease at San Francisco General Hospital and the Director of the UCSF Infectious Disease Fellowship Training Program. He is also co-chair of the Infectious Control Committee and chair of the Antibiotic Advisory Committee at San Francisco General Hospital. Dr. Chambers has published over 100 papers and textbook chapters in the areas of drug resistance, bacterial infection, and staphylococcal bacteria, staphylococcal diseases. His clinical and research interests are antimicrobial disease resistance, staph infections, and experimental therapeutics.
Thank you. It's great to be here. In the next few minutes, I'm going to start from a clinical angle, but weave science back and forth as we go through the talk. I welcome you to interrupt if you want to do so, if it's okay. I don't know what the rules of engagement are, but I have no problem with being interrupted. I deal with medical students, and they're an unruly bunch, so it's not an issue. I'm surprised so many people would come to hear about such a dry topic. The only fun word in here, I guess, is kill, and then we have bacteria, of course, to work with. This is quite an old disease, and I just wanted to share with you the first case reports of staphylococcal infection, which in fact was in Exodus. It is not well recognized that the sixth plague of Egypt was in fact an outbreak of staphylococcal disease, and it's, the citation is, so they took soot from a kiln and stood before Pharaoh, and Moses threw it toward the sky, and it became boils breaking out with sores on man and beast. And that is just a great description of what the disease process is for most people who acquire staphylococcal infection. You'll also notice if I go through, even though the title is Methicillin-resistant staph, I'm going to speak largely about staph in general because I think that is the real issue. The methicillin resistance question to me is a transient phenomenon, although important at the time, and I think I will show you why I think that is the case, and clearly I'm not trying to underestimate it. But I don't want to distract from the overall problem of methicillin-susceptible and methicillin-resistant disease. So we need to go back in time to look at this relationship between Staphylococci, Staph aureus, and humans. You can see that staph were here well before we were. The ancestor of this organism dates back 1.6 billion years. Biologically, the evolution of all staphylococcal species, and there are over 30, is very closely related to the emergence of warm-blooded animals, primarily mammals, but also birds. So there are Staph aureus equivalents in all animal species. It's just the Staph aureus, for us, is our human pathogen. And we have been co-evolving for many years. Depends on how far back you want to trace this lineage, but I think we can agree that humans have been here about 250,000 years, and in that time, Staphylococci have been co-evolving. And here's Moses. You can see how late the case reports were. We know it was going on back here as well. So just a quick primer on what Staphylococci are. It is a genus designation, so it is Staphylococcus genus, aureus, like Homo sapiens. The defining features are it's gram-positive and it's in clusters. Here's a picture of them up here looking under the microscope. They're quite tiny. That's a couple of microns across. Gram-positive is just a staining feature. They stain blue if they're positive, red if they are negative. They're very hardy, these organisms. You can leave them in the environment and they will do just fine, thank you. They have an extensive accessory genome. that is acquired by horizontal gene transfer. And this is one of the key messages that I want to communicate to you is how this biology works. I've told you the lineage is 1.5 billion years old. There are 30 different species. It co-evolved with all warm-blooded animals. Now, its natural habitat are mucosal surfaces, the nares, anterior nares, the nasopharynx, skin, axilla, groin, perianal area, non-intact skin. Staphylococcal species, broadly writ, are the most common organism on all of our bodies. We carry normal staph that are colonizing and do not cause disease in most cases. So Staphylococci are mostly commensal. Even Staph aureus. In this very room, there might be 30 people. 10 of you have Staph aureus right now somewhere on your body. Sorry, it's just the way it goes. It's unavoidable. So there are a lot of people who have Staph aureus. 90 million people in the United States right now are colonized. A proportion of those will have methicillin-resistant Staph aureus, but they never get disease. And so this balance between the evolution, the adaptation of the human and the co-selection with Staph aureus is really quite profound when you think about the biology. So it's highly human adapted. Asymptomatic carriage I've mentioned. There are 90 million people in the United States right now who are colonized. 5 million are colonized with MRSA. It is spread by direct contact. This doesn't float in the air. You have to physically touch the organism to acquire it. Or you have to physically touch a wound that is infected with the organism. Or you have to be in close contact with somebody who is a carrier of the organism. It has the capacity to be quite virulent, and I'm going to show you some photos that are really not great for lunchtime, but that's when I was asked to talk as we go through the clinical manifestations of this disease. And it has many, many virulence factors. Many classic bacteria, such as plague or anthrax, have 1 or 2. You knock those out and say virulent. With Staphylococci, there's no single virulence factor that you can eliminate to reverse the course of disease. And it has a remarkable capacity to become drug resistant. Let me just say a word about the host defenses. Primarily, it is an intact skin. So the barrier to staphylococcal infection is no breaks in the skin. It's hanging out in your nose. It's happy to hang out in your nose, but if you get a cut, that presents an opportunity for a portal of entry. There are antimicrobial molecules that we make called defensins that are effective in low counts of bacteria at eliminating organisms. And then a white cell response, PMNs, polymorphonuclear leukocytes, neutrophils, are responsible for mopping up staphylococci. And here we have some staphylococci that are in the process of being ingested by this neutrophil. And here's this neutrophil trying to get a hold of those 2 cocci there that he's not been able to catch up with. Now, so that's our host defenses against staphylococci. Do not think for a minute that staphylococci are not working on the same problem from the other side, right? They want to avoid all of this. And I just want to show you some examples of this, of targets that humans present in terms of their host defenses. So I mentioned the antimicrobial peptides. Well, staphylococci make staphylokinase, other molecules that eliminate or inactivate specifically these peptides, specifically the human forms of these peptides. There's chemotaxis, that is when the poly comes in to eat the bacteria. There are specific inhibitors of that process, so the poly is paralyzed. Red blood cells may aggregate the organism. These are lyses to provide iron nutrients. Oxygen-mediated killing, you just go down the line that whatever we throw up as a defense, staphylococci have been working on for the same period of time they've been hanging out on us or before. So this is not a passive process, it's incredibly dynamic. In terms of the mechanism of disease, there are 4 primary diseases that staphylococci cause. They cause local infections, that would be an abscess or a cellulitis. There's food poisoning that's not truly an infection. It's, I don't know what you're having for lunch, but if the mayonnaise has been sitting out a little bit too long, you might be susceptible to that. It incubates in food and then causes a preformed toxin which makes you sick. Invasive disease and then toxic shock syndrome, and I'll just show you examples of each of those quickly. Abscess and cellulitis, I think you can see this, but here's this individual thigh, the red area, you see this punctate thing in the middle, I think. It's probably about this big. That's filled with pus. That's what an abscess is. It is a pus-filled collection that is an isolated and focal infection. This is fairly benign as things go. You can lance it, people will do fine. They may recur, but most diseases of this type, because the host is able to contain the infection. Here's another example of a lesion that is more cellulitis. There's no drainable pus here. It's a little redder and angrier looking. This is the classic spider bite of MRSA. It is not caused by a spider at all. It is just a focal infection that is very inflammatory. Food poisoning. So it causes intense nausea and vomiting and explosive diarrhea. And the point of this whole slide actually was to set up this picture right here, which I found on the web of. So it's short-lived. He's not going to be here long. Lasts about a day or two. No fever. You get over it. Self-limited. But if you're debilitated, it can make you sick. Invasive disease. So hold on to your cookies. This one's pretty benign. This is a patient that we saw in July last year. Now, July is not normally influenza season, but you know last year with H1N1, it was influenza season. And this is a patient who was seen in the outpatient setting and had H1N1 influenza A. Was seen in the outpatient clinic. I know you're not used to looking at chest x-rays, but in this situation, black is good. This is the heart, so this is a normal chest x-ray. Guy shows up, and I think you can appreciate that now we're seeing white in there. What that is, it's pus in the lungs that have accumulated in less than 24 hours. This patient went on to die of a lethal infection, appeared only to have influenza when he showed up. So he's an at-risk person whose host defenses obviously did not work. At the top, these are heart valves. Doesn't matter which ones, but you can see that there's this gluck on them, we'll call it that. What that is collections of bacteria and clot on the heart valve. Normally a heart valve should have a nice smooth surface like you see here. You can see how ratty this looks, and you can see that there is this excrescence called a vegetation that is on the valve that breaks off, and it is loaded with bacteria, and it lodges in the circulation. In this case, you can see this foot here, and these are petechiae. They're micro blood clots and abscesses from this thing breaking off and spreading throughout the body. This is a brain scan in which you can see where the white spots are, where these clots have embolized and lodged in the brain. So it is a truly overwhelming systemic illness. The other disease that is common in people who have staphylococcal infection is osteomyelitis. This is an MRI, which I understand has finally been scheduled for Tiger Woods for today. How it can take Tiger Woods like 3 days to get an MRI, I don't know. But anyway, here we have a spine. This is a neck. I think you see the arrow here, and then on this projection, you see that white thing in the back. What that is a collection of pus and destruction of the bone that is encroaching on the spinal canal. And finally, this is called necrotizing fasciitis. This is a patient who had a skin infection that became invasive and deep. In order to get rid of it, he has to have all of the skin and soft tissue debrided. Okay, so these are unusual, rare forms of the disease in the big scope of things. Not so rare when you pull all invasive disease together, but for most of us, we'll never have this type of infection. Finally, toxin-mediated syndromes. This is like a bad sunburn with shock that is toxin-mediated. This is toxic shock syndrome, in which the hand is desquamating. I think you can see that the skin is beginning to scale off. There's a redness. It looks like a sunburn. This is a toxin-mediated disease. I think you can see these purulent pustular-containing lesions about the size of a dime. These are caused by staph that make a toxin, which cleave the epidermal-dermal junction. So you get a blister just like you had a burn, but it does it chemically and enzymatically, not by heat. And here's a systemic manifestation of that in a patient who has overwhelming sepsis. This is a toxin-producing strain in which this EKG lead was moved from here to here and denuded to the skin as it separates off. You could actually touch this person and have the skin scale off. Here's an infant syndrome called scalded skin. So that's what Staphylococcus can do. In terms of the types of infections, if you look at all Staph aureus infections, 90 to 95% are skin and soft tissue infections, SSTI. So only a minority are these more serious infections, and I've shown what they are here. The most common organ systems are the blood, the urine, the respiratory, the bone, and the joint, but it can affect literally any organ in the body because of this ability to spread through the bloodstream. How much staphylococcal disease is there? Well, we really don't know because there's no prospective surveillance to measure this for us. We can get at it backhandedly by looking at cases and then doing prevalence studies in certain communities. So what I'm going to show you here are not hard data. These are best guesses from indirect means of measuring Staph aureus disease. This is not MRSA disease. This is the whole picture, okay? So 90 million people already told you are colonized. In terms of boils, abscess, skin and soft tissue infections, 1 to 3 million a year. Now that seems like a lot to me, and it is a lot. It gives you an attack rate of about 1 to 3% because colonized people are more likely to get an infection. So this is a fair burden of disease. But in terms of the severity of disease, relatively easily dealt with. What we need to worry about and attracts all the attention is the invasive disease, of which there are 150,000 cases a year. Roughly half are MRSA, half are MSSA. 70 to 80% is healthcare associated. You do not have to be an MRSA strain to be healthcare associated. If you say, okay, do MSSA strains, that is susceptible strains, cause healthcare-associated disease? Yes, they do. And they cause the same burden of healthcare-associated disease as MRSA. It's just they're not drug-resistant. There are 30,000 deaths annually. If you happen to get one of the infections, the odds of you being rehospitalized with a similar problem is about 25%, and it recurs about 20% of the time. So, turning then to what is methicillin-resistant Staph aureus, how does it differ? And as I said, first and foremost, it is Staph aureus. It is, often labeled a superbug. I don't know where that term, actually I know where it came from. It came from some scientist trying to get funded, probably. Because he says, oh my God, we have a superbug, we need to destroy it. And it's really not a superbug any more than MRSA or MSSA is able to infect humans. It's just drug resistant. It is resistant to the beta-lactam class. Now that's really important because the beta-lactam class is far and away the most effective antibiotic. So penicillin, methicillin, those are the drugs that are used, other similar drugs. Cephalosporins, carbapenems, the entire class. Most type of resistances do not take out an entire class, but this type does. That is the concern with methicillin resistance. And it accounts for 50%, maybe slightly more, of all invasive infections. So it's about half the disease burden. Now, how do they do this? Here comes the science part. So there are two elements we have to think about. Excluding the host. Remember, while all this is going on, infectious disease is so green, I love it. You got the ecology, you have the host, you have the environment, you have the bacteria struggling to live, you have the host fending them off so there's no infection. It is so dynamic. And in the middle of all of that, there is a ton of gene exchange. And the two elements from the bacteria point of view are the chromosomal core genome. That is, its genetic representation that is common to the Staph genus, not to Staph aureus necessarily, the Staph genus. And then you have mobile genetic elements. The common chromosomal core genome contains housekeeping genes. These are not virulence genes usually, although there are some in Staphylococcus that are called innate. This toxin, staphyloxanthine, makes Staph aureus. Aureus is golden. It kind of has this yellow property. On agar, that accounts for that. Coagulase coagulates blood. Catalase is a host defense mechanism. These were acquired millions of years ago as part of mobile elements. You can see the footprints of them, but now they're part of the core genome. The more recent acquisitions are on these mobile genetic elements, and they carry two types of genes, resistance genes and virulence factors. Where do the virulence factors come from? Well, they're acquired probably as modifications of other staphylococcal genetic elements that are imported into Staph aureus and that are adopted by Staph aureus and provide an advantage to them against the host. So that selective pressure is the host. The resistance genes, where do they come from? They're entirely a creation of antibiotic selection, entirely. Staphylococcus in its natural form has no drug resistance, zero, none, nada. It is susceptible to every single drug. So all the drug resistance that we see has totally been acquired. So this is what the core genome might look like. It is circular, in fact, much teenier. But when you examine the core genome that is the genus genome, and you look at it at a sequence-based level, there are these little islands peppered all over it. And what are they? They're mobile genetic elements that have been acquired by the genome and integrated to give an advantage to get by. There are also plasmids that are extrachromosomal. These are free-floating genetic elements that can come and go. They tend to carry drug resistance factors. This is just a listing of the number of mobile elements. There are insertion sequences, prophages, transposons, cassettes, integrated plasmids, free plasmids, pathogenicity islands that contain drug or virulence factors that were acquired over time and integrated in the genome. All the drug resistance that we see has been acquired in the last 70 years, but the mechanism is the same. Now I want to spend just a couple of minutes upon why antibiotics are an agent of selection. How does this work? So the simplest theory is that the organism or the gene, and I say theory, it's really not a theory. I mean, this is what happens. You can demonstrate it easily in the laboratory. There's an organism or a gene. In our situation, it could be either, right? We can select the organism or we select a particular gene. Remember, we're talking about both. And that there's a spontaneous mutation. There's nothing the antibiotic does to make the mutation. It just happens because the enzyme that makes DNA makes a mistake in about 1 in every million nucleotides. So 1 in a million, there will be a mistake in the gene. It may be silent. If it's lethal, it's eliminated. But if it causes no problem or an advantage under the selective pressure of an antibiotic, there's clonal expansion and then amplification as it slowly becomes the predominant strain if it has an advantage of the mutant over the parent. The easiest way to do this, when I say spontaneous mutant, it could be a mutation like an error in the DNA, but the easiest way is to acquire foreign DNA, because then I don't have to figure it out. I have my buddy over here who knows how to be penicillin resistant, and so all I have to do is import that resistance element into my own cell, and this happens, and then be selected, and then I have clonal expansion. So that's what is going on. Horizontal gene transfer drives most of this, and this is so important to realize because we keep our eye on the strain, but we forget we're selecting the genetic element. It's easy to constrain a strain theoretically. It is much harder to control a genetic element that is horizontally transmissible. So in this example, this is how penicillinase resistant works. Penicillinase resistance is a narrow type of resistance. It only destroys penicillin. All the other drugs that I showed you, their beta-lactams, work. It is on a plasmid. The origin of the plasmid is not known for sure, but probably some other related species in the genus. There are mechanisms by which it's horizontally acquired, primarily by a viral infection of the cell. Yes, bacteria have viral infections too. If they survive it and there's foreign DNA in the virus that infected them, it will be taken up, and if it offers an advantage under selective pressure, it will survive. So you can see only one of this number of cells has it here, but under selective pressure, in this case an antibiotic, you can see it's the lone survivor. And then as part of vertical gene transfer, all of its progeny may carry this plasmid, and they will be drug resistant too. Not shown in here is that they can back donate this plasmid to anybody up here as well. So you got two things going on here, the plasmid and you have the cell. Methicillin resistance is another type of horizontal gene transfer. But this time the gene is not on a plasmid, it is integrated in part of the chromosome. It is actually integrated like a cassette, and it is a cassette. So here it is in this donor. This probably, wouldn't you know, came from a rat staph strain years ago. It was integrated into a Staph aureus and other related staph strains as well. Taken up by the chromosome under selective pressure, vertical gene transfer, same deal all over again, okay? So methicillin resistance works by not destroying the drug, but by altering the drug target. So this is fundamental, it's a better approach. If I want to destroy the drug, I gotta have an enzyme that destroys every single drug. If I got 50 drugs, I need 50 enzymes. But if I have a target that is changed, that entire class, if it depends on that mechanism, will not be effective, and that's what happens here. This element is quite amazing. It's called SCC-MEC, staphylococcal chromosomal cassette. MEC is for methicillin resistance gene. This cassette element doesn't have to carry drug resistance. It can carry lots of stuff. But this one just happens to carry drug resistance. It always integrates in the same place in the chromosome. There are enzymes on it, the job of which is to integrate it into the chromosome or to excise it. This is the transmissible form, but it is not self-replicating. Plasmids self-replicate. They don't have to be in the genome to replicate. This is a genome parasite. If it does not integrate into the chromosome, it will die. So it wants to be in the chromosome, but it must excise itself to be transmitted. Okay? Now there's a size restriction on these things. So methicillin resistance actually dates back to the 1960s in London. Prior to that, there was penicillin resistance that I told you is plasmid-borne, and on the x-axis is the timeline, the y-axis is increasing resistance to beta-lactams. Within really a few years of penicillin being introduced, there was penicillinase resistance. It was interesting to go back and read those reports because the descriptions are just like methicillin resistance now. They go, oh my God, it's resistant, but it's just in the hospitals, don't worry. And they go, oh my God, it's gotten virulent, and moreover, it's in the community. And that was this particular lineage of strains Foch type 8081, penicillin resistant. Methicillin was introduced, and this MEC element was invented. It was actually remodeled from another Staph strain. It's called SCC-MEC type 1. This is not a very good element. It's a prototype Model A version. It doesn't transmit well. It has a mistake in its transmitting enzymes which make it stuck in the background, so that these strains had to spread strain by strain by strain. The resistance could not spread by moving the element because it's immobile and trapped in the genome. Over time, they fixed this problem. And there are two other elements that were invented. These are remodeled versions that bear structural similarity to the original version, but now these are a 1930s model. Their problem was they're too big. They're too big because they carry a lot of extra drug-resistant stuff because they were born and bred in a hospital environment. But over time, they are able to spread. So this strain that carried this element died out, does not exist anymore. And it was replaced by these more efficient strains that were still in the hospital that slowly increased in number, and this was the outbreak of methicillin resistance that occurred worldwide now in the 80s and 90s. Note the increasing resistance. MRSA4 is the prototype of modern drug-resistant MRSA. It has a streamlined, stripped-down element that only carries methicillin resistance. It exacts little or no cost by carrying other drug resistances. That's good because other antibiotics work, but that comes at the cost of transmissibility. This element is able to spread like wildfire. It's rather like the plasmid element that contains the penicillinase-resistant gene. So under the current dynamic, these strains are dying out, and they're being replaced by these strains, both in the hospital and the community. And by being replaced by the strains, the element is surviving and transmissible and able to create new strains, as well as the strains in which it was already resident and spread in hospitals and the community. And this just shows you the continued outbreak as we had to rely on other narrow classes of drugs, of drug-resistant strains. Now I want to briefly go over evidence that isn't much discussed for a phenomenon that is probably just as important for drug resistance. Remember, in the first model, what happens is there's a spontaneous mutant, and then if the drug happens to select for it and it's beneficial, you'll get a mutant. This one, the drug causes the mutation, then selects for it. So here's how this works. We have the organism or the gene. A mutagen, that is something that makes a mutation, is applied. There's a variant that is under selective pressure and you get an evolved gene. Antibiotics do this. They cause stress in the bacteria. They make the bacteria take up foreign elements. They make mutations occur because the stress generates certain enzymes in the bacteria that generate mutations. So the mutation frequency is increased 10 to 100-fold over what it would be, and it's no longer spontaneous. It's generated by the drug. So this phenomenon is only recently recognized as being important in this business of drug resistance. And again, in this model, the mutagen and the selecting agent are the same. So I think I will conclude there and take questions, but first, Staph aureus is highly human adapted. Normally, it doesn't cause much problems. It has become human adapted through acquisition of horizontal genetic elements that are integrated into a core genome. The period of selection of those elements has been the periods of hundreds of thousands of millions of years as they've had to deal with the host. Antibiotics are kind of a host factor, but they're quick poison for bacteria and highly selecting. And all of this is driven by antibiotic selective pressure. Not just the bacteria, but the elements as well. These mobile genetic elements are key for virulence factors, they're key for antibiotic resistance, and the ultimate driver of the antibiotic reservoir is virtually limitless because you're talking about the pool of core genomes and the mobile genetic elements which can be mixed and matched in any way conceivable under selective pressure. So those are my prepared remarks. I hope that gave you some insight into how remarkable this pathogen is. And you know, people who study bacteria, we secretly admire what they do. You know, we never turn totally to the dark side, but you have to appreciate how adaptable they are in order to contrive means for interdicting what it is that they might do. So thank you for your attention.
Some of you may know I'm Rush Holt. I represent Central New Jersey, and I'm a co-chair of the Biomedical Caucus. I wanted to thank Dr. Chambers for the presentation, which is, as those of you who attend these sessions can attest, is very much in the tradition of excellence of these presentations. There are a couple of points, actually one question that I wanted to ask to lead off the question. The point I wanted to make first is, I imagine you and most of the people in your members' offices think that, well, of course, this research goes on in fighting infectious diseases at NIH. Well, only if we maintain it. That's the whole point of these presentations. It is absolutely critical that we maintain it, and not at a subsistence level. This leads me to my question, and then guessing at what your answer will be, I'd like to ask make a comment on the basis of it. Whether it's by plasmids or cassette integration into the hardware of the cell, I think the evolutionary advantage given is really pretty small. So if we take away this preferred environment of the drug, it will return to the less virulent, it would be overtaken by the less virulent forms of this. Is that?
Well, virulence maybe not.
Well,
drug
okay.
Resistance perhaps.
Virulence, okay. Virulence maybe not, but drug resistance perhaps.
Right.
Okay.
I think that fundamentally, this is a Jekyll and Hyde problem with Staph aureus, because most of the time it's colonizing people in this room and you'll never get an infection, but something happens. For example, remember I said you break the cutaneous barrier. The first thing that happens to you when you go to the hospital is your cutaneous barrier, among other things, is broken, right? You get an IV put in, you get a Foley, They cut you open, all kinds of stuff happens to violate that most critical defense. And so the opportunist and virulent staph lurking is then able to take advantage of that situation. Then the drug selection is yet another feature of the process. So if you withdraw the selective pressure, then yes, over time there will be no advantage. It just depends on what the pressure is.
Yeah. Well, and so the environment that provides this selective pressure favors one strain, and there isn't you've removed the community's corrective mechanism in effect. I would argue that we see the same thing in research areas. If we force areas of research to scale back so they don't have a widespread, vibrant, self-correcting community in research, we end up with pathological research. So it's to make the point that I was making earlier, we must maintain a vibrant, well-funded biomedical research program in the United States. In order to have the kind of the many-faceted research that is necessary for the broadly useful outcomes. So anyway, thank you very much.
Yeah, I'm not going to say no to that, you know.
Let me let you take the other questions.
Thank you very much.
Yeah.
Hi, you said healthcare associated. I assume that means hospital acquired. And then I have another question.
Yeah, so I should have clarified, I'm sorry. Healthcare associated means an infection that occurs in association with contact with the healthcare system. So that does not have to be hospital acquired. That is the best example of it because there's no doubt of the acquisition. If you go into a hospital and you're perfectly normal and you get surgery and you get a surgical site infection, that is a hospital acquired infection. Now I should point out that the bug that caused it may not be hospital acquired because if you're a carrier, you may already have it. Okay, so there's those fine distinctions. So you don't know whether it's hospital acquired because you acquired the bug in the hospital or whether you had the bug. So you have to be careful about those relationships. But the infection is, I like the term hospital onset infection. It's unambiguous. Hospital acquired kind of makes you think, well, you know, it's all the hospital's fault. And I'm not trying to whitewash hospitals, but the microbial dynamics are much more complicated than that. Healthcare associated means, let's say you go in the hospital and you're fine, you have surgery, but a month later you have an accident and are readmitted to the hospital and you get a staph infection. That would be an associated because you were in the healthcare system. So that's how you count them. Community onset then can be healthcare associated or community associated. But when you start looking at all the staph disease, it is centered around the healthcare delivery system when you look at invasive disease. And why is that? It's because of the skin barrier and those things that are done in hospitals that invade and violate, if I may use that term, the host in terms of their natural host defenses. I mean, that's what happens when you go in the hospital. And to the extent that you do that more and more in the population that undergoes those type of treatments grows, you can see why an organism that would be just hanging out in your nose might cause trouble when there's a medical intervention.
And then I just wanted to ask, is this one of the bugs, is this one of the situations that's exacerbated by all the antibiotics used in factory farming, or is that different bugs?
I hate to say, probably not staph. But the gene pool, for many resistance factors, that are not so important now can be traced to gram-negatives, which is what you're talking about, other bacteria. And it just, anytime you start dumping antibiotics, this is going on in the background level all the time. And the genetic factor that might be useful for me today, I do not own, but some other bacteria is working on it, if you see what I mean. It's just so important to understand the horizontal transmission mechanism of perpetuation of drug resistance.
David Miles, I'm working with Congresswoman Rosa DeLauro. And at the beginning of your talk, I was intrigued that you said this notion of resistance is transient. Is it transient because of intrinsic factors of the microbes themselves that they're continually evolving, or is it because we have more drugs in the pipeline? I thought it was paradoxical because after my time working in the hospital, I mean, we run out of drugs. I think Banco is now the thing that we use now to treat MRSA, and then we're having some resistance to that too. So I'd be curious if you could unpack that.
Yeah, transient is probably too mild a term. I would say it's conditional based upon the selective pressure that you apply. That'd be a better term. Because the situation is so fluid and there's so much adaptation. So if we quit using vancomycin today, there would be loss of vancomycin. You can show it in the laboratory. You can take a vancomycin, a strain that's resistant to vancomycin, and you throw it into the laboratory, and you start a little race, and you take the vancomycin susceptible guy and the resistant guy, and you say, okay, grow. And the susceptible guy beats out the resistant guy every time. But pretty soon, the resistant guy starts throwing off back mutants that now grow, and they catch up with this. So and that's all in the absence of drug pressure. So that's what I meant to.
As a tool for your research, how important is genome sequencing and the advances we're making in rapid genome sequencing? Is that important
Huge.
To getting there from here?
I'll give you a concrete example. So an experimental antibiotic, I won't go into details about it, but it's an experimental antibiotic, was being developed. And so I was asked to evaluate its ability to select for resistance, and we started in the laboratory because it's being developed. And so we made a number of genetic constructs that would represent the possible array of genes that would be under the pressure of this antibiotic. Now, how did we make those constructs? Well, we had the genome sequence. So we knew the targets, we went in, we engineered it, and we were able to make the tools in order to test the thing, okay? We test the drug, and, somewhat in a boring fashion, the targets that we thought would be important proved to be because they became mutant. But we also did a control strain in which there were none of these targets. And it became resistant too. So we go, very interesting. And the first thing I did, I accused the postdoc of screwing up, of course. This can't possibly be right. So what did we do? We knew the strain that we started with. We knew the mutant. We rapid sequenced them both. And within 2 days, we knew exactly where the mutations were in that strain. And now this allows us to be able to go back and work backwards and figure out how this new type of resistance works. So
if because we're dealing with really the worldwide competitiveness of the United States in maintaining a leadership position in rapid genome sequencing for a lot of different reasons. If we're able to develop tools that would allow you to do the sequencing twice as fast, 10 times as fast, et cetera, is that going to have a big impact on your work in trying to deal with this resistant strain problem, or is it like a 1% improvement in your ability to?
I think the whole technology, I can't answer whether twice is, if I have to wait 4 days instead of 2. But when you get into volumes and trying to plan experiments, it's not just do you get 4 days instead of 2, it's now I can do 100 variants instead of 10. Right. That's where the calculus comes in and works in your favor. Right. So it is tremendously important. It also has practical applications in terms of diagnostics. I didn't talk about that, but one way to get around this whole drug resistance problem is don't be slopping antibiotics on bacteria that don't need to have those particular antibiotics. And how do you tell? Well, you can sequence them. So there are practical applications.
So how would that story have been different without your ability, without the advantages of genome sequencing?
How would I have done the experiment?
Yeah.
So you have to take this mutant that you don't know how it works, and then you try and you use one of these, you exploit the horizontal gene transfer phenomenon, and you put in this promiscuous element that will integrate all over the chromosome. Okay, they exist. And then you try and knock out the resistance. The problem is, when you do that, it might wind up in another part of the genome that also lowers the resistance, but for some other reason, because you've taken out an important protein. So it's guesswork. It's not directed. It would take, I don't know that we could have figured it out, to tell you the truth, because we're talking about single mutations concentrated in targets.
Any idea how long that would have taken you to do?
Well, I'm not sure it would be doable in the final analysis without some sequencing. It's just hard to imagine how you, because it would be high risk, because you would have to count on that approach landing in the very gene of interest and in no other. And still you got to do sequencing, because you have to go in and see what gene you're in. And if you didn't have the genome, you wouldn't know what gene you're in, right? So it really does build up in terms of a critical database. And the more sequencing, the better, you know, as far as I'm concerned from the biology point of view, the science of biology. But not only sequencing, you got to have you have to have the biology to back it up.
I was wondering if you could clarify a little bit more back to the food animal question that was raised. If I understand correctly, is it true that really the issue is that there are different strains of MRSA and different ones that are causing different levels of illness in the United States so far? So for example, there might be a healthcare associated strain that is primarily what we're most concerned about right now in the US, and that does not appear to be associated with livestock. But there is also a separate livestock strain of MRSA that does seem to
the pig one.
emerging as linked to antibiotic use in food animals, though in the US we haven't seen as much illness caused by that yet, although in Europe.
And here's how they're linked. They're linked not by the fact that the pig bug is in humans. They're linked by the cassette element, which is mobile. Okay? So the pig bug got infected with a resistance element that is broadly shared in the population. That's what happened. See, that's why it's so important to understand about these horizontal elements, because, you know, it could be way over here and not even in the world of Staph aureus. But if it happens to come in contact because it's been born and bred in the right environment and provides an advantage, if it can integrate and be taken up by Staph and provide an advantage, it provides an advantage. I also want to say that don't ignore the community MRSA problem. The hospital strains are, you know, you don't want those either. But the community strain, hospital strains are a little weakened. You know, they're not quite as virulent because they're hanging out in hospitals. So they have to be drug resistant. So this causes a problem for them. Community strains that are beefed up, virulent, and they're drug resistant. Right, so, and it's the whole population of Staph. That's why, you know, Staph aureus. I don't care if it's methicillin-resistant or methicillin-susceptible. The only problem with methicillin-resistant is harder to treat, right? So that's an issue.
This is all fascinating as far as understanding and learning the science, but from a public health perspective, does it just mean that we will always be, instead of being a year ahead, then we'll now be 15 minutes ahead of knowledge? I mean, are things going to keep changing? You'll learn quicker, but the bugs are going to be changing quicker. And do we just need new types of antibiotics?
Yeah. So are you talking about the sequencing, the technology that allows us to learn things faster?
Yeah.
Well, yeah, I don't know that those are related competing issues.
Well, and also just clinically, we're understanding how things are evolving,
but it suggests a way forward, and you can test whether that's liable to work. What it for example, the laboratory science revealed that antibiotics not only select for resistance, they produce resistance. Now that is profound. So once you have that piece of information in hand, then it makes it doubly important to be more responsible about your antibiotics. In terms of antibiotics, I don't know if you've seen the press, but you know, the last one that was approved, well, it was last year, telavancin for gram-positives. The pipeline is just, now it's not even a pipeline. There are just not very many drugs. And there are lots of reasons for that. For one thing, the first discovered antibiotics were easy, and they were easy for a reason. The later ones are hard for a reason because we already got the easy ones, right? Also, the biomedical, or the community is less willing to tolerate a toxic drug, so a lot of those type of issues come into play, which make it more important to preserve the drugs that we have. So.
Two questions. With the mobile genetic elements, is it understood what sort of elicits them to excise out of the genome and go into another? Is that like some sort of environmental pressure? And then the second question that I have is, do these mobile elements have a signature such that you can sort of track their movement through various strains? So if there's a mobile element that's common in hospital-acquired infections, right? I mean, how can you distinguish that, for instance, that particular mobile element didn't get into agriculture, multiply in there, and then sort of become prevalent in agriculture, and then sort of infects, transfer to another strain that, and so then it looks like it went from agriculture to person, but perhaps it went from hospital to agriculture back.
You can't know the ultimate origins of the element. But what you can know is, if you sequence enough strains, that it's not in those strains, that it's only in the human isolates. What you don't know is where did the progenitor come from? And that's what we need to think about too. The element that you see now may not have been the element that got this rolling. In fact, people think that, and it may have some other advantage for the strain in its normal resident background, that when you now put it in a virulent strain and apply some antibiotic pressure, it mutates. There's no rule against it mutating that either. It remodels it, so it just provides tremendous genetic potential around the model of just having one super bug. If it's just one super bug, you got to spread it by direct contact. If I have one super mobile genetic element, then I get both ways. So, and it's the origins that you can't know about. But the element probably came from normal flora on humans or animals that were drug exposed, probably humans. Ultimately, this element that I'm talking about. It's probably in the normal flora that are the Staph, not aureus. That is where it came from.
You made a great point about how it's not just about MRSA, and you spoke broadly about Staphylococcus. Looking on the horizon, are there any other bugs or bacteria, you mentioned gram-negative, that you see as being like other big threats, like moving forward? Because people are so focused on MRSA. What other bacteria?
Well, you've already put your finger on the gram-negatives. And what worries me most about bacteria, although when you look at the burden of disease by these rare drug-resistant ones that get all the attention, and those are important, I don't want to minimize them because they represent a reservoir of resistance. But what's really bad is when you get common bacteria that become drug-resistant. So like, for example, E. coli. E. coli is in everybody's bowel. It causes urinary tract infections. It's 90% of the urinary tract infections in women, E. coli. It's like third or fourth on the hit list of bugs that can get in your bloodstream. Again, it's a commensal, right? So it doesn't, under the right circumstances, no problem. But if it's drug resistant, there are antibiotics that I used 10 years ago that I can no longer count upon for somebody who comes in and they've never had an antibiotic because it's now part of the resident flora because they've acquired this element as well, and that's what's happening in the gram-negatives. So I would have to say the gram-negatives. Now the ones that get all the press, you know, the biodefense bugs, what a waste of time and money. I know it's not popular to say, but those are not the problem. You know, understanding their biology is interesting, and I'm not saying we should ignore them. But, you know, how many people died of anthrax in the last 10 years? 7? How many people died of MRSA? 30, or not, of Staph aureus, not MRSA. 30,000 last year. That would be 300,000 in the period of, you know, I mean, it's just, anyway. So I think it's the more common organisms that we need to be worried about because they have the public health implication.
What if there was a major aerial attack with a anthrax strain that had been engineered to defeat Cipro and Doxy? Then would that change the picture?
Well, certainly if there were. Then I would say to you, though, shouldn't we work on preventing the aerial attack? Because I don't know about the drug resistance part. And understanding what other drugs work is certainly important. But I'm talking about just the burden of disease and what the likelihood of that is. You have to look at the common organisms because they're the problem. I mean, you know, let's just balance 300,000 people with the whole aerial attack. That is more a tool of terror than an implement of healthcare disaster in a certain way, to me. I mean, it's, you know, I don't know how many people, I'm sure you probably know since you asked the question, how many people. And I'm not for aerial attacks with anthrax. I want to go on record. I want to go on record. I want to go on record for that. I probably was a little extreme. I tend to drift off into that anyway. But the comment was just look at the proportion of where the problem is, and then the competing risks and where you're going to spend money when you're looking at healthcare-related research.
What about the importance of having a vaccine against Staph aureus and a vaccine against anthrax?
Good. That would be good.
In terms of not having a resistance issue.
Yes, that would be good. Both.
Well, there is a vaccine against anthrax.
No, I know.
Oh,
Yeah.
okay.
Yeah.
I'm just I'm talking about vaccine approach as opposed to just drug therapy.
Yeah. Yeah.
How come only 30% of the population is colonized? Is that they figured out?
That is a very good question. No one really knows. There are interesting phenomena about it. So when you're born, if you culture teeny babies, about half are colonized in the first month. And then that slowly goes down, and then it blips back up in adolescence to about 30 or 40% colonized, and it slowly goes down again. So at 30% is an average figure. So there are clearly factors that control that. Otherwise, why would they clear, right? There are also certain risk factors that make it more likely that you're colonized. And one of them is, for example, diabetics who take insulin. Now, is it because they take insulin or because they're diabetic, or is it because they use a needle to administer insulin? Who knows? But they're more likely to be colonized. If you take people who have skin diseases, they're more likely to be colonized. And that kind of makes sense because the skin is so important as a barrier. So you start monkeying with the barrier, then Staph aureus is kind of able to hang around. One place it cannot be is on normal skin. I could paint you with Staph aureus. Do not try this at home. I could paint you with Staph aureus and come back tomorrow, and as long as I did it on normal skin, I'd recover no Staph aureus. Your coagulase negative, the normal ones, they would run it out of town. And whether it's because the skin contains molecules that inhibit growth or whether the other bacteria, we didn't even talk about that, but other bacteria kind of don't like Staph aureus moving in the neighborhood. And so you eliminate them, then Staph aureus goes wild because, you know, the residents have moved out, and the next thing you know, you've got a crack house in your neighborhood. Same thing, you know.
Say colonize, you mean inside
Outside, yeah. But nobody really understands the biology of why people are colonized. And the interesting phenomenon is if you're colonized, you're more likely to get a staph infection, but you're less likely to die of it than if you're not colonized. So there might be, getting around to this immunization question, there might be some advantage if you don't have an antibiotic of being colonized. The cost is, you know, you get a pimple, but the benefit is you do not die. Easy choice. Unless you're a teenager. I guess that pimple or dying is like, they're the same, right?
Dr. Chambers, thank you very much.
You're welcome.